Hello everyone, let's talk about the DNA. So DNA stands for deoxyribonucleic acid. It is a molecule that stores genetic information in all living organisms. It has the instructions for building proteins, and these are going to determine the cell structure and function, and also your physical appearance, ultimately. DNA is found primarily in the nucleus of eukaryotic cells, and they exist as these long coiled polymers molecules called chromosomes. And even though DNA is incredibly long, it is actually efficiently packaged and highly organized within the cell. In prokaryotic cells, however, we don't have linear DNA molecules like we do in eukaryotic cells. In prokaryotes, the genome is much smaller, so the DNA molecule is in the form of a little circle. So it's basically a circular DNA molecule. So if we take a look at the structure of the DNA, we're going to see that it is a double-stranded molecule, and the two strands intertwine to form a double helix. And each strand is actually a polynucleotide. Why? Because it's made up of many repeating units called nucleotides. So basically, these nucleotides are linked together into chains. So I'm going to go ahead and circle one nucleotide for you. So here you go. Also notice we only have 4 different nitrogenous bases in the DNA, so adenine, thymine, guanine, and cytosine. And the sequence of these bases is going to determine your genes. It's going to determine what you are going to develop into. Are you going to be a plant? Are you going to be an animal? Are you going to be a fungi? Basically, all species, they only have those 4 bases, and yet the sequence of these bases, that's all it takes for you to have variations within the species. I find this incredibly incredible and amazing. So now let's look at what the DNA monomer, which is the nucleotide, looks like. What does it look like? If we look at it, we will see it consists basically of 3 components. So we have a phosphate group, and the phosphate group consists of phosphorus surrounded by 4 oxygen atoms, and we know that these oxygen atoms are going to be electronegative, so we'll have a negative charge. And this is the reason why DNA naturally has a negative charge, because this phosphate group is sitting in its backbone. And the next component is deoxyribose sugar. We can also call it pentose because it has 5 carbons in its molecule. And then nitrogenous base. So nitrogenous base could be any of the 4 bases: adenine, thymine, cytosine, or guanine. So now what we want to do is make sure we remember why we numbered the carbons. We'll learn that in the biochemistry unit, but let's go ahead and revisit this. Notice your sugar, which is pentose, has 5 carbons, and we need to be able to count those carbons in order to understand the directionality of the DNA molecule. So your base, notice, will always be attached to the 1' end. So this is the carbon right here, and we're going to call it 1' end. And then once you find your base, remember, you just go clockwise and count all these carbons. So this will be your carbon number 2. We're going to call it 2' end. Carbon number 3, 3' end. Carbon number 4, 4' end. And carbon number 5 is off the ring because we only have oxygen atom right here. So the carbon number 5 is sitting right there. So as I said before, nitrogenous base is going to be attached to always 1' end. 3' end is going to be another end that's important because it's going to offer a functional group, a reactive part of the molecule, which is OH group. That's your hydroxyl group. That part is going to be engaged in the chemical reaction when we are linking up the nucleotides to build a strand. So it's going to be involved in dehydration synthesis. And then the carbon number 5, 5' end, notice will always have a phosphate group attached to it. And this is why this end of the molecule is going to be referred to as 5' end, versus this end of the molecule, which is that sugar part with the hydroxyl, is going to be referred to as 3' end of the molecule. So now let's take a look at how do we actually form a nucleotide. So if we take that sugar molecule, your deoxyribose, and a nitrogenous base, notice your sugar offers hydroxyl group at this prime end, and nitrogenous base offers hydrogen. So when these two come together, we form HHO, which is really your H2O. So that's the water molecule that's going to be released during the dehydration synthesis. And the bond is going to form between this nitrogen and this carbon. And you can see this bond has formed right here, and that's your glycosidic bond. So whenever you have nitrogenous base and the sugar that have combined, we had actually formed a structure that we call nucleotide. Now, if we add the phosphate group, we're going to form a nucleotide. So how do we add the phosphate group? Notice your sugar molecule offers hydroxyl, and the phosphate comes with the hydrogen at this position. So once again, we have HHO, that's your water molecule, H2O, that's going to be released, and the bond between this carbon and this oxygen is going to form. What do we call this bond? Phosphoester bond. So if we make one more, guess what we're going to have? We're going to have what's called phosphodiester bond that's going to link up those nucleotides. So now we know that DNA is not a single-stranded molecule. It's a double-stranded. But what does it look like when we link up those nucleotides? We get one strand. So you can see one nucleotide is already enclosed in this box. I'm going to go ahead and draw another one, circle it for you. Phosphate, sugar, and the base. Here's another one, phosphate, sugar, and the base. And phosphate and sugar is going to make up the backbone of the DNA molecule. And then notice nitrogenous bases is going to connect those backbones. And if we look at the molecule again, notice one strand of this molecule will have a 3' end, and the opposite will have your 5' end. So now how do we build the second strand? What do we have to do? We have to apply base pairing rules. We mentioned before that we have 4 different bases: adenine, thymine, guanine, and cytosine. So adenine and guanine are called purines, and they will have 2 ring bases. So notice right here, this is my adenine. Here's ring number 1, number 2, so there are 2. And if you look at pyrimidines, we have thymine and cytosine, so these are going to be only 1 ring bases, so just 1. So how many rings do go across the DNA? What do you think? What would be the width? Yes, if you said 3, you are correct, because we would be 1, 2, 3 rings that go across, right? So we're going to build this uniform DNA ladder. So now how do we pair up those bases? Adenine pairs with thymine because they can form 2 hydrogen bonds. So here's your bond number 1, number 2. And cytosine pairs with guanine by forming 3 hydrogen bonds. 1, 2, 3. If you have a DNA molecule with a lot of guanine and cytosine versus another DNA molecule with a lot of adenine and thymine, which molecule do you think would have a higher melting point? If you said guanine and cytosine, the one that contains lots of guanine and cytosine, you are correct, because you have 3 hydrogen bonds in between to break. So it means it would take more energy to be able to break these bonds. And this type of molecule would be more stable. So if we put everything together, we're going to construct a DNA double helix. And notice the strands run in the opposite direction. So we're going to call them anti-parallel. So if this end of DNA molecule is your 5' end, here's your phosphate group at the 5' carbon end of the sugar, and this is going to be your 3' end of the molecule. And then the other strand looks like it's flipped upside down, so this is going to be your now 3' end, and this is going to be 5' end. So the DNA, the strands look like they run in opposite directions. So if we had to build the DNA molecule, we would go from 5' end to 3' end and add the incoming nucleotides to the 3' end. And then the other strand, notice we're going to run in this direction from 5 to 3 and add incoming nucleotides to this prime end. Now I know right now it might look like you're not sure how this could actually happen, but I promise when we get to DNA replication unit, you're going to see how it all fits together. So another thing I want to quickly mention is that, remember we said nucleotides are linked into polynucleotide strands, and that's the process of dehydration. And we're going to form that phosphodiester bond between the adjacent nucleotides. So if we take a look at the incoming nucleotide, we have hydrogen at the phosphate group, and then this is your hydroxyl group. We know this was your 3' end of the molecule, and water is being released. This is why we call it dehydration or condensation reaction. And the bond between this carbon and this phosphate is going to form. And then if we go back and we look at the entire bond, because remember this was your phosphoester bond, so this bond right here and this bond right here that was previously formed, the bond between the 3' end and the 5' end of the other nucleotide sugar molecule with the phosphate group in the middle. That's your phosphodiester bond. And the last thing that I wanted to add is that when we combine all these features, looking at the structure of the DNA, which was, by the way, described by Watson and Crick, DNA resembles that twisted ladder with sugar phosphate backbones on the outside and the base pairs on the inside. This structure allows the DNA to be stable, compact, and capable of accurate replication. Now, mistakes do happen. We call them mutations, but overall, the process is very accurate. I will see you next in DNA replication.